Assalamualaikum. Today I'll talk about lectine or MBL pathway of complement system. MBL means menin binding lectin. There are three pathway of complement system activation. The first is classic pathway. The second is alternative pathway. And the third is lectine or MBL pathway. In previous video, I talked about classic pathway and alternative pathway. As I mentioned there, the classic pathway requires immunoglobulin M and immunoglobulin G. And in alternative pathway, it requires bacterial endotoxin, which is lipopolysaccharide, and other substances like snake venom or yeast cell wall. And in lectin pathway, it binds with the polymer of sugar called mannose of bacteria. Those bacteria which contain mannose on their surface can initiate MBL pathway. Suppose it is a bacterial cell wall. Suppose this bacterial cell wall contains mannose on their surface. Mannose is a polysaccharide. Suppose here is the mannose, and also it requires mannose binding lectin, which is a protein from liver. Here is the liver. Liver synthesize complement protein C1, C2 and up to C9, and also lectin binding protein. Liver synthesize and release menin binding lectin, and they are all the time circulate in the blood. And whenever mannose containing bacteria enter into the body, the MBL protein bind with their mannose. MBL are the protein and mannose are the polysaccharide, and MBL bind with the mannose and form a complex which later bind with the C4 complement protein. C4 complement protein fragmented and become C4b and C4a. C4a is the smaller fraction and it releases into the blood, and C4b remain attached to the mannose and menin binding lectin protein complex. Then C4b bind with the C2 complement protein and divide it into two parts, C2b and C2a. C2a release into the blood and C2b bind with the C4b. Now C4b and C2b, both of them act as a C3 convertase. And they act on C3 and divide it into C3b and C3a. C3a release and C3b bind with the C2b. Then C4b, C3b and C2b act as C5 convertase and act on C5 and divide it into two fragments. The larger fragment in C5b and smaller fragment is C5a. A release into the blood and C5 remain bind with the other fragment of complement protein. Then C5 act on C6 and produce C6b, C7b, C8b and C9b. And C5 to C9 called membrane attack complex. They attack the membrane of the bacteria and make pore in their membrane so that the water and electrolyte enter into the cytosol and the bacteria begins to swell and ultimately it will burst and the cell or bacteria will die. On the other hand, the C3A and C5A act as anaphylotoxin because they act on mast cell and release histamine, which is an inflammatory mediator. That's all about lectin or MBL pathway of complement system activation.